All right, so last little thing to discuss as we're looking at cells and what's going on here is we're going to talk a little bit about cell communication. A lot of times you might refer to this as cell signaling. It's really how cells can interact with one another and really have certain cells influencing what other cells might be doing. One of the things I do want to say in this one, I know we have a wide range of students that are taking this course. Anything from honestly students that have already had cell bio to some people that have just had bio and chemistry. So in terms of how far and how much depth you want in here on these ones, it can be very distinct. I do not want this to be kind of a cell biology level of information. We have a course for that, which I don't teach, and I don't want to teach it. So on this one, again, this is more of an introduction to how different cells can communicate with one another, and I'm going to introduce some of the things that maybe in another class you might talk about in a lot more depth. Something I might ask is just kind of how much, in terms of how much I expect you to know, is just kind of that these are things that exist. Like in terms of receptors, I'm going to give you an example of a few different receptor types. I just want you to know that these are things that exist and are some of those types. I'm not going to ask you specifically about how they work differently. So again, why do cells have to communicate with one another? I mean, really it comes back to the idea, and I've said it once or twice already, we are a multi-celled organism, and in terms of number of cells, we have a ton of cells. The number they put to it is about 38 trillion. So the amount of interconnectivity is important there, and for certain cells to continue to do what they need to, a lot of times they're going to have to be influenced, or basically you're going to have to receive information from other cells. So this lets other cells regulate what other cells might be doing, allows other cells to tell us what an external stimulus might be. We'll see as we get to the heart and other things like that cells need to communicate with one another, or with nerve cells need to communicate with one another, or endocrine, for example, the number of organs sending signals to other cells, the endocrine system, are all ways that these cells are communicating with cells that they might not be connected to. And if cells aren't able to communicate with one another, a lot of times this can lead to dysregulation of different systems, and that can be a problem. So understanding how cells communicate does end up being kind of important to us. So again, there is different types of signaling between cells. There's different types, 3 general types of chemical signaling. So in terms of this, we can have ones that are directly cell to cell. So this is actual contact, something like a gap junction. The second one is the gap junction. We could have cell-to-cell signaling in terms of a receptor or something holding, like with immune cells, holding a cell out here and another cell interacts with that one. We can have connections between cells like heart and cardiac muscle cells. The other thing we can do is have pretty much substances being released, be it a hormone or a neurotransmitter, which in reality are very similar things, being sent from one cell to cells that are maybe closer or much further away. The main thing on a lot of these stuff here is we're going to be dealing with stuff that is called a ligand. A ligand is just the particular substance that's going to bind to a receptor. So when we talk about a ligand, it is what the ligand for a particular receptor is what's going to bind to that ligand. So you see the ligand is going to be what binds to that receptor, sorry. So this chemical signaling is most of what we have going on here. So most of the cell signaling in the body is going to be this chemical signaling. These are going to be chemicals a lot of times that, it can happen a number of different ways, but a lot of times they're going to be freely diffusing from between cells or entering into the bloodstream and traveling to other cells. Sometimes this could be chemicals that are directly from one cell to the cell that has contact with, so it's like that cytoplasmic stuff and the signaling going through that. But again, these are pretty much some type of chemical traveling between cells. In order for a cell to do stuff with this, it is going to have to actually be able to receive that signal and then do something about this. So a lot of times we talk about this idea of kind of there's 3 steps of cell communication. This idea of receiving an extracellular signal, transducing that extracellular signal, so taking it from what it was, the original signal, and this multi-step process that might happen within the cell. And it doesn't mean that the actual substance is traveling through the cell, but it's going to be, might be a signaling cascade that was activated by this particular ligand that's going to send this down to the cell, and then we bring about some type of cellular response. Might be a change in gene level or transcription. Could be a change in how the cell is actually working, other things like that. So we're going to have to worry about each of these, and we're going to kind of talk about these each in a little bit of detail. So this is again showing you that idea. You can see on the side of the screen here, you have reception. You have some type of ligand binding to a receptor. Transduction is taking that signal from here, because this ligand might not be able to enter and go directly to bring about the response. It might be what we call a second messenger system, where it has to bind to a membrane receptor and then get signal down a pathway in order to get it to where it needs to bring about its particular response. So reception, again, obviously kind of ends up being important here. Usually is going to involve a membrane protein. So this is usually the part with the reception we're talking about. We have some type of membrane protein that a ligand can bind to, and that is what's going to initiate this whole signaling process. We're going to see that there is different types of these ones. I'll give you some examples towards the end of this. Transduction is then taking that signal and basically changing it. So originally it was this particular hormone or chemical on the surface. We're going to convert the signal into something that brings about a cellular response. So it might be one type of enzyme altering the next one, that sooner or later gets to where we have a response. And we're going to see a lot of times there is a large number of steps involved in this. If you ever take cell biology, you'll see these signaling pathways and the ridiculous complexity that there is in them. A lot of times there is a large number of steps. But usually you have something that attaches to that plasma membrane receptor, and then this is going to start moving them down this path of signaling molecules to bring about a response. So this shows you an idea here of like a particular signaling molecule. Again, don't worry about there just being an I channel and a G protein and all this stuff. I don't care about that stuff and you knowing this stuff at this point. It's just kind of getting this idea of that here's the reception, binds to a particular hormone here, and this whole step here to get to that cellular response, these are all the different steps to that cellular transduction process. Where, okay, the G protein might have to go over here and interact with this ion channel to open it up, and then these ions flood in, and this brings about a change which activates a sensitive protein that brings about a response. The main thing is we change or transduce this signal to this response. And then finally the response, it really depends on everything that it's connected to and what's going on here. The response to cell signaling can be very, very different depending on the type of cell and what's going on here. Might be turning on an enzyme, turning off an enzyme, increasing transcription of a gene, decreasing transcription of a gene. These are all things that can happen in response to cell communication. And again, really is going to depend upon the protein and what that protein is connected to. And this is one of the things I just want to kind of point out. You can get the same signal. So for example, the signal of adrenaline or epinephrine is the other way we say adrenaline. There's different receptor subtypes of this one. These different receptors, an alpha receptor and a beta receptor, can have completely different responses. So I mean, to give you one quick example, vasodilation and vasoconstriction. You can get vasoconstriction of a lot of blood vessels and then vasodilation of blood vessels in the muscle and around the heart in response to epinephrine. Same signal, bringing about exactly opposite responses in different blood vessel types based on the receptor and the transduction pathway that's present. And this is again showing you this. The other thing here that is also important is one signal can a lot of times lead to more than one type of cell response. And we're going to see this a lot of times is because there is these things like amplification and branching of these pathways. Again, stuff that we're not going to delve into the nitty gritty of it, but we're at least going to introduce these ideas a little bit. But again, one signal can bring about a lot of different changes from a single ligand. So this is kind of showing you a ligand binding to a receptor and the change here. But one of the things that happens a lot of times with this one is each of these steps, a lot of times one protein might activate multiple proteins. So a lot of times what we see with these signaling pathways is we see an amplification of that signal. So an amplification and a branching or divergence of that signal. So again, this is showing you how the ligand here catalyzed one change here. That wouldn't be much of an amplification. But a lot of times we're going to have these multi-step pathways where initial stimulus might activate one thing, which activates the next one, which activates the next one, till we get to the actual response. And a lot of times at each of these steps, there may be an amplification of that signal. So, for example here, you might get one receptor binding here, but this might be able to activate 3 proteins here, which activate 3 proteins each down here, which activate 2 each down here, and you can see how that signal can get much bigger from that single signal at the beginning. So this is that idea of that amplification or divergence of the signal. And again, just showing you a couple different times here with some different systems, how you can get each of these activating multiple things, and again, can get a much larger cellular response from a very small initial signal. Again, a lot of this ends up being very important in that signal reception. And again, to me, a lot of times the response is going to be very much dictated by the receptor and what's attached to it. So that signaling molecule is going to bind to that receptor that's there and usually bring about some type of change in that receptor. And that's how we receive that signal. So ligands, like I was saying here, depends on the ligand, and we'll see as we get into the course a little bit more. If we talk about testosterone, estrogen, progesterone, these are what we would call lipophilic signaling molecules. They are lipid-based, cholesterol-based hormones. A lot of these things are capable of going through the membrane and binding to receptors inside the cell, where a lot of our proteins and other type of signaling molecules have to bind to cell surface receptors. So a lot of times we talk about ligands needing a second messenger system or being able to enter directly into the cell. Certain things can go directly all the way into the nucleus as well to bring about their change. So not everything's going to go through a pathway, but a lot of stuff that we will talk about would. So that's why I'm just introducing the idea. So steroid hormones, like I said, are those lipophilic ones where, or things like nitric oxide, where a lot of these other ones you're going to need this cell surface receptor. So what I wanted to show you is just a few of these different receptor subtypes. So there is some different ones here. If you ever take a cell biology course, you'll get into a lot more detail on these ones, but these are some of these kind of second messenger type of systems. So you have what are called G protein based receptors, tyrosine kinase based receptors, ion channel based receptors. And again, I just want you to know that these are things that exist. I'm not going to say with an essay, give me the difference between a G protein versus a tyrosine kinase receptor, but I do want you to know that these are some of the ones that we discussed. So I mean, this is showing you a G protein one here. Meaning in this one you have a membrane bound thing here, and a lot of times we call this a second messenger system because you have a membrane bound protein that is going to be able to take an extracellular signal, get it through the membrane to the inside of the cell, which can then bring about different changes inside that cell. That's an idea of a G protein based one. Tyrosine kinase based receptors, this is something I worked with in my graduate school work. These don't use G proteins. These are a lot of times dimers, so you have 2 different parts of the receptor at the surface of the membrane. When the ligand binds, they connect to one another and you get this phosphorylation cascade that happens. So I can show you right here. You have a signaling molecule bind here. The 2 parts of the receptor come together. It causes the tail to be phosphorylated, which can phosphorylate other signaling molecules, which tend to pass these phosphates to other signaling molecules until we get to some type of response. And again, that's a tyrosine kinase receptor. So, and again, don't worry about the different steps involved here, but again, just trying to show you an example of this. Ion channel receptors, we will work with these a little bit when we talk about nervous system and the muscles and how muscles contract. These ion channel receptors, a lot of times will have a molecule or a signal that causes them to open. The binding to the ligand tends to open up this channel and let ions that have a particular concentration gradient to move in or out of the cell, like we were talking about with diffusion and osmosis, to move in or out of the cell based on the concentration gradient. We see this a lot with muscle cells. We pump a lot of sodium ions out and potassium ions in. See it with nerve cells as well. When the particular neurotransmitter binds to that particular ion channel, it can let those move with their particular concentration gradients, and it can lead to a change in the cell, which can lead to a cellular response. So that would be an example of an ion channel receptor. And again, just giving you a few different ideas of cell reception. So you can see here, some type of signaling molecule binds to this, causes the channel to open up, lets the particular ion or substance travel in or out of that cell based on its concentration gradient. So, I mean, that's the basics of kind of the cell signal reception, transduction, and kind of response. What I want to talk about now is just some of the different ways that we signal both locally and longer distance within the body. So you can get some of these different types of signaling here. So paracrine signaling is just this idea, and we don't have autocrine on here, but sometimes the cell can release a substance that turns around and acts back on the cell. That is called autocrine, on the cell that released it. Paracrine is a cell releasing a hormone or some type of neurotransmitter or some type of ligand, let's just say, and it can act on the cells that are very close. Again, any cell to receive the signal is going to obviously have to have the receptor, and that, again, is the key thing here. Anything with signaling, you always have to have the receptor if that cell is going to be able to have a particular response to those particular signals. So synaptic signaling, again, it's a similar idea. We're releasing a chemical. Again, it's just a short distance here, where long distance signaling or hormonal or endocrine signaling, we can have a cell that releases its particular signaling molecule into the bloodstream, which can then travel and end up binding to different target cells found in different areas of the body that have the particular receptor. Direct contact, these are again going to be gap junctions, so we see those a lot in cardiac muscle. It lets ions from one cell move into another cell. We also see some of this with the immune system in the cell-to-cell recognition, where you have a particular surface protein that can bind to a particular surface receptor on a different cell. So a lot of times with T cell and B cell signaling, these T and B lymphocytes in the immune system, we can see this type of stuff happen. So again, the long distance stuff here, you might have a particular neuron, a particular endocrine organ. It can release its substances into the bloodstream. These are going to travel throughout the bloodstream. They're going to diffuse out into that interstitial fluid. The only cells, again, that are going to respond are those that have a particular receptor. Again, different receptor subtypes might lead to different cellular responses. So one particular signal doesn't always mean the same response in every cell that has a receptor. You can get different receptor responses based on different types of receptors that combine this. So like I said, with epinephrine, you have alpha and beta adrenergic receptors, and you have different subtypes of those. Same thing with acetylcholine. You can have some different cell receptor types that can lead to different responses based on that, and the signaling molecules in the transduction pathway. Again, we can get different responses from the same signal. So again, this is showing you, again, this endocrine cell, releasing that, traveling through the bloodstream, getting to a particular target cell, and bringing about a response. Again, all this just ends up being important because as we get throughout this course, this is just introducing the idea of it. I'm not expecting you to understand everything about every body system that can do different types of signaling and what goes on with them, but this is a way to introduce these ideas of cell communication before we talk about the endocrine system, before we talk about neurotransmitters and the nervous system or muscles and acetylcholine and how the nervous system communicates with the muscular system. These are all just ways to kind of introduce this idea and give you just kind of the basic framework of it. Again, obviously for anything that's endocrine or neuroendocrine, we need the bloodstream involved in it. And again, autocrine is if the cell releases something and it acts directly back on itself. Paracrine is going to be on these neighboring cells. Generally, we start saying endocrine if it's traveling a distance in the bloodstream. So like I said, aero and autocrine's back on itself, paracrine to neighboring cells. And examples of this, histamines, different inflammatory cytokines are good examples of this. Interferon, another good example of this. We'll talk about that in AP2 when we talk about virally infected cells. I mean, local diffusion and the idea of a neurotransmitter across a neural synapse is a similar idea. I mean, in ways, it's not way different than what the endocrine system is doing. It's just a much shorter distance that it's signaling. I always use the example of this being a lot like a walkie-talkie, where if you ever get those little ones for like when you're hiking, the ones that need line of sight, that's basically what's going on with a neurotransmitter. Short distance to send the signal across, where endocrine is more like a radio broadcast. Like right now, there is a radio broadcast going, but because I don't have my stereo on, I can't hear the broadcast that's going on. It is being broadcast. I don't have the receptor active right now, so it's not getting the signal. Again, doesn't need bloodstream for this. And again, you can have these local regulators, things like neurotransmitters or other things like histamines, some of these small signaling molecules that are very much local regulators. Hormones, we generally start referring to hormones are ones that are going to end up in the bloodstream or the lymph and travel throughout the body is really kind of the big difference between those.